There we go. And thank you very much, everyone, for coming along. Okay, so today I want to talk about how the ocean can change its biogeochemistry in ways that may lead to carbon release. And to explore this, I'm going to look, as we do in my research group, at events in the past, which might help us better understand processes that could occur in the present and the future. So today, arguably, the ocean is one of our greatest allies in the fight against climate change. It currently takes up around 1/4 to 1/3 of anthropogenic carbon, takes up a lot of heat as well. And our models of future carbon uptake by the ocean, as shown in the right-hand side, tend to predict a fairly persistent, smooth uptake of carbon by the ocean, mitigating CO2 change. However, when we look into the paleo past, we see that in some instances, the ocean has been a much more dynamic driver of changes in CO2 and climate. And this, I think, is important to consider as we enter a warming world where we have the possibility of abrupt and unexpected changes. So I want today to highlight two potential modes of change that have occurred in the past and we might want to keep an eye on in the future. The first are modes of ocean CO2 release during disruptions of the Atlantic meridional overturning circulation that occurred during the last ice age. And the second is centered on changes in calcification and calcification feedbacks that may occur in an acidifying ocean. So we're going to begin with this record of abrupt climate changes of the last glacial cycle. These were first most famously observed in Greenland, and I'm showing the record of oxygen isotopes from Greenland in this top panel. The abrupt warming and cooling events that you see here are of the order of, these are jumps here of the order of about 10 degrees C, and they occur in the space of about a decade. So these are really pronounced and abrupt shifts in climate. Now, there's still a variety of debates about the exact mechanisms that occurred during these events, but almost all of these center on changes in North Atlantic circulation and sea ice, due to abrupt reorganizations of the overturning circulation. Now, it would be possible to give multiple talks on just that circulation process by itself, but that's not what I want to discuss today. I want to discuss the consequences of these circulation changes for biogeochemistry, the topic of this forum. And we see that pretty pronouncedly in terms of what happens to atmospheric CO2 during these times. So you can see that during these periods of cold over Greenland and in the North Atlantic, associated with a reorganization and likely a weakening of the overturning circulation, we can see these centennial scale increases in atmospheric CO2. And so the question that I want to address to begin with is what causes CO2 rise during these AMOC disruptions? So if we're thinking about CO2 change on order 100 to 1,000 year timescales, a really critical place to look is the ocean, and in particular the Southern Ocean. The reason for this is that the circumpolar channel round the Southern Ocean allows, in coupling with the westerly winds, allows the upwelling of deep carbon-rich waters. And when these when these upwell to the surface, the inefficiency of the biological pump in this region means that much of this CO2 outgasses to the atmosphere. So for a long time, hypotheses for how to change CO2 on glacial, interglacial or rapid centennial type timescales have focused on changes in partitioning CO2 between the deep ocean and the atmosphere in the Southern Ocean. And in the last few years, we've been fortunate to be able to generate some paleo data from deep sea corals, which allow us to track this relatively directly and some of the processes that may lead to this occurring. So the first example of this I want to show you is based on the boron isotope composition of deep sea corals, which acts as a proxy for seawater pH. I'm showing these data here plotted upside down such that carbon-rich, low pH waters are up and carbon-poor, high pH waters are down. So what we found in these data is a record of deep Southern Ocean carbon storage, which looks like the mirror image of the atmosphere. During the last glacial period, we find low pH carbon-rich waters at depth in the Southern Ocean, evidencing enhanced deep ocean carbon storage. And then over the deglaciation, in a series of steps associated with those cold events in the North Atlantic, we see CO2 release from these waters and rising pH as the deep Southern Ocean gives up its carbon to the atmosphere. And you can see these corresponding steps in atmospheric CO2. So why does this happen? Well, a wide variety of mechanisms have been proposed, but currently two of the most discussed are changes in sea ice, or the loss of sea ice as the deglaciation proceeds, in part enabled through redistribution of ocean heat due to those cooling events in the North Atlantic. That sea ice loss is shown by this proxy from Antarctic ice cores in blue. So you can see sea ice loss in these blue events here corresponding to those atmospheric CO2 rise events. And the other, I think, most currently discussed mechanism is a poleward shift in the austral westerlies. There's a reconstruction of the westerlies position here in pink. And again, you can see the way that is coupled with the change in atmospheric CO2. And I think it's important to point out that both of these phenomena, a loss of Antarctic sea ice and a poleward shift in the westerlies, are both occurring today, and so should give us some pause for thought, some cause for concern perhaps, in terms of the future of the Southern Ocean CO2 sink. Might that change indeed from sink to source, or at least might that current day CO2 sink weaken in the future? Now, superimposed on the centennial scale changes in CO2 that I've just discussed, we also see these fascinating, abrupt, multi-decadal jumps in CO2 that I'm pointing at with these arrows here. And these are of the order 10 to 15 ppm CO2 rise within 50 or so years. Now, when these were first published with very new kind of high-resolution ice core data, a variety of people thought, oh, is this just too fast for the Southern Ocean to be able to do? Do we have to invoke some faster-moving part of the carbon system? Is this due to the terrestrial biosphere, say? However, with our deep-sea coral data, we're able to record snapshots in time at the resolution of these kinds of jumps in CO2. And again, we find corresponding changes in Southern Ocean circulation, though of quite a different and unusual nature. So we're looking again at some of this deep sea coral data. I'm zooming in on the CO2 jump during the cold period at the start of the last deglaciation. And now I'm showing decay-corrected radiocarbon age, which gives a sensitive tracer of ocean ventilation. Now, today, Southern Ocean waters have fairly old radiocarbon ages. They're not very well ventilated. We can get some kind of sense of context for that if we look at a modern distribution of ocean radiocarbon ages. So Southern Ocean deep ages are order 1200 years or so, because there's limited air-sea exchange, which is required to kind of reset and reventilate the radiocarbon tracer. And so it's really quite extraordinary that during this interval of CO2 rise, we see this very pronounced ventilation event where deep sea corals in the Drake Passage down at depths of 1700 meters jump way above their glacial ventilation state, and indeed way above their modern ventilation state, to reach radiocarbon ages of around 400 years. So that's like a 400-year offset between the atmosphere and waters at 1700 meters depth in the deep ocean. And again, looking at the modern ocean for context, the one place today that waters that deep get this close in radiocarbon age to the atmosphere is the high latitude North Atlantic, and that's due to the vigorous ventilation in this region, in particular via pronounced convection and deep water formation. And so our best mechanism to explain this pronounced jump in ventilation at this time is the onset of invigorated convection in the deep Southern Ocean in this window. And this is indeed something that has subsequently been seen in a variety of climate model simulations. So I'm showing here some simulations by both Laurie Menville and Matteo Villet. And they showed that in response to, again, one of these cold events, disruptions of the AMOC in the North Atlantic, you can see invigoration of convection in the Southern Ocean, shown in this blue stream function here. And when this occurs, the ventilation of this water, which was previously carbon rich, leads to a rapid increase in CO2 in the atmosphere, as we observe in the deglacial CO2 record. Now, the Southern Ocean can clearly, as we've discussed, go through some quite exciting different modes, but this is not actually the only region that has a say in the biogeochemistry of the ocean and the atmosphere at this time. So I also want to highlight the role of the Pacific Ocean and the North Pacific Ocean in particular. An important reason to look at the Pacific Ocean when we're thinking about the carbon cycle is nicely illustrated by this Google Earth view here, which is just that the Pacific is absolutely massive. We live on a blue planet. We're 2/3 ocean, and about half of that ocean is the Pacific. So the Pacific by itself has about 30 times the CO2 of the atmosphere, and it really matters what the biogeochemistry of the Pacific does in terms of controlling atmospheric CO2 on these timescales. So again, over the last deglaciation, we've looked at radiocarbon in the Pacific, and this now is data from both benthic, that's kind of deep sediment dwelling forams, so they're living on the sediment surface, in this case at 3,600 meters from a sediment core. I should say here, sorry, time is going now from right to left. And then here is the radiocarbon age of the planktonic forams living in the surface water above that core. And what we see at the start of the last deglaciation is, again, this dramatic collapse in radiocarbon age, where the deep ocean and the surface collapse towards one another. And the best explanation of this is, again, the onset of deep convection, which we find in a variety of models occurs in response to shifts in atmospheric rainfall and redistribution of ocean salinity. In these models, again, because we take what were previously old carbon-rich waters and we expose a bunch of that water to the atmosphere, we see dramatic loss of carbon from that water. That's shown in these colors in here in a simulation by Laurie Menville, and in some of our own simulations, we show that when we successfully recreate that ventilation event, so the ventilation data is in the stars, some model simulations of that in the lines, this leads to pronounced increase in atmospheric CO2. Okay, so to summarize this first part of the talk, during past AMOC disruption events, we see an increase in atmospheric CO2, which can occur both on millennial and kind of centennial to decadal timescales. Now, the CO2 release can occur as a result of sea ice loss and a poleward shift in the westerlies, and also on faster timescales in response to the abrupt onset of Southern Ocean convection. And we also see an abrupt convection event in the North Pacific, which again acts to release carbon from the deep ocean to the atmosphere. Together, I think this highlights the interconnected nature of ocean circulation and biogeochemistry, and how different basins and their biogeochemistries can be interconnected, can interact, and lead in some to quite dramatic changes in atmospheric CO2. Okay, so in the final 5 or 10 minutes of my talk, I want to touch on, or 5 minutes, I want to touch on a very different kind of biogeochemical response to a changing climate. We're going to look at calcification in an acidifying ocean. So to remind you, ocean acidification, sometimes known as the other CO2 problem. This is the fact that the uptake of CO2 into water releases protons, making water more acidic. And this decrease in pH has potential impacts on calcifying organisms. However, if we want to actually look at records of ocean pH, they're fairly short-lived. They're about the same age as I am. Read into that what you will, maybe kind of just about old enough to learn something from, but you know, it would be no bad thing to have that bit much more experience of our changing ocean pH. And this does make it hard to distinguish forced change from multi-decadal variability. In addition, while some culturing and mesocosm studies show pronounced impacts on calcification from acidification, sometimes the results of these kinds of studies can be a bit mixed. And there's always the worry that when we come to think about calcifying plankton in particular, there are multiple generations of some of these organisms within a year. And so on the kind of 100-year timescales of ocean acidification, there's a legitimate question to ask about the potential for adaptation and adaptive responses. So to address this, we wanted to look at reconstructions of ocean acidification over the historical period. And I'm showing some new boron isotope-based pH reconstructions from 3 high-resolution cores just here. So these are 3 places where we have nearby instrumental data that's shown in the black line. And now we're going to extend this back to about 1700 with our boron isotope reconstructions. And so from this, you can see that the records show a fairly consistent signal, a close match to the nearby instrumental data. And when we combine these, we see a, if you like, kind of hockey stick curve for ocean acidification, an upside down hockey stick, showing about 0.14 pH unit acidification compared to the pre-industrial, with a close match to atmospheric CO2, including both relatively early onset of acidification. You can see that by even the early 1800s, we are notably acidified compared to the 1700s. And that, in fact, tracks that very early rise in CO2 due to land use change. And then more dramatically, we see this acceleration of acidification from the 1950s onwards. So this, in a way, is perhaps a picture that we have all in some ways imagined in our heads, but was nonetheless important to reconstruct. But what was perhaps less expected was, well, what does the response in calcifiers look like? So we've traced that as well using size normalized weights on a couple of different species of planktonic foraminifera. And what these data show are that for these different species, in around the kind of latter part of the 20th century, we see a decline in calcification by order 10% in Beloides and 25% in G. ruber. And this suggests that there is really already a visible and pronounced effect on calcification in these key planktonic calcifiers. And this has important implications both for ecosystems and their functioning, and also indeed for the future of the alkalinity sink. Now, finally, I want to deal with an example of ocean acidification that we find deeper in the geological record. And this reveals a different and somewhat surprising potential long-term response of calcification to a carbon cycle perturbation. So we're going way back in time. This is the PETM, the Paleocene-Eocene thermal maximum. It's a hypothermal event. I'm actually going to deal with a slightly earlier hypothermal event. It's called the latest Danian event. It's a bit like the PETM, if you've heard of that. If you haven't heard of any of these, the thing to take from this is that it's a time where we know that there's an input of carbon into the ocean atmosphere system that's tracked by carbon isotopes. This is from some reduced, some organic source. Now, in response to this input of carbon, we can see acidification of the ocean, which is tracked in a variety of different metrics I'm showing here. And we also see warming in sea surface temperatures. And in some ways, all of that is kind of expected, right? We put carbon into the ocean atmosphere system, we get acidification, we get warming. The thing that was unexpected is that when you look at this in detail, we see that although that kind of carbon input seems to dissipate, the carbon-13 recovers to kind of pre-excursion values, the carbonate system, so ocean pH and indeed sea surface temperature, does not. They stay stuck in this more acidified and warmer ocean state. So what might be going on here? Well, to dig into this, we wanted to look at a variety of things that could potentially alter or be influenced by ocean carbonate chemistry. And we were struck by a surprising response in key calcifying plankton. So if we look first at the foraminifera, this coarse fraction metric traces the weight of calcification, the weight of foraminifera, so gives a broad measure of the extent of calcification. Now, what we see at the onset of this event is perhaps the kind of expected response. This is the same as what we just looked at for the industrial era. We see this kind of classical acidification response, a decrease in calcification, a decrease in shell weights. But what happens next is unexpected. You can see that then over tens to order 100,000 years, we see this increase in coarse fraction, representing a rough doubling in the weight of foram shells. And this is associated with a kind of evolutionary shift from these thin-walled pre-muricate taxa to these much larger and more heavily calcified morozavelids. And I've put these two pictures on the same scale. And we also see a striking and somewhat similar response in our calcifying nanoplankton. So our coccolithophores show roughly a 1.5 to 2 times increase in cell volume. So there's a pronounced increase in calcification. What might the impact of this increase in calcification be? Well, to step through this, I want to start by thinking about something that I think most people are reasonably familiar and comfortable with, and that's the idea of the biological soft tissue pump, whereby the uptake of CO2 by, say, diatoms fixes carbon into organic matter and thus removes CO2 from the surface ocean and the atmosphere and stores that at depth in the deep ocean. But when we're considering changes in calcifiers, we also need to consider the role of the hard tissue pump, which is sometimes called the carbonate counterpump. And this is because through the net uptake of alkalinity, this tends to decrease pH and increase CO2. And so if we were to increase calcification with bigger, thicker, heavier calcifiers, we would expect to strengthen the alkalinity sink, strengthen this hard tissue or carbonate counterpump, lowering surface ocean pH and elevating CO2. And indeed, that is what we appear to see in this event. Now, why might such a shift take place? I don't know the answer to this. I'm maybe happy to speculate on this a bit in the questions, but I will just briefly flag that one possible reason for this is that this may be a response to a pulse of alkalinity coming in during this global warming event that may favour more alkalinity hungry calcifiers. And I think that that is a real concern as we think about potential enhancements to silicate weathering or ocean alkalinity, in that the work done by this has the potential to be undone by this calcification feedback. Whatever the exact cause, the net result is that we see prolonged acidification and warming. Okay, so to summarize, we see in our recent reconstruction acidification apparent by the 1800s, accelerating in the latter half of the 20th century. We can see already an impact on calcifying organisms, but the long-term future of alkalinity is going to depend in large part on the planktic calcification response, and there can be a variety of different possibilities there. If calcification decreases, we end up with a stronger CO2 sink. that if calcification increases, we end up with a stronger CO2 source. And I think this underlines how important it is to keep an eye on ocean biogeochemistry in a changing world. Okay, thank you very much. Sorry for running slightly over. I'll look forward to questions in a wee bit.
Okay, thank you, James. Thanks for that. It was fascinating. So we hand over to our second speaker, Seb Henning from the University of Edinburgh.
Hello, everybody. Today I'll be talking about predicting loss of cold water coral habitats and a little bit about our work going from in situ to ex situ and then into the in silico modelling approaches. I'd just like to start with this picture. So this is a picture which I took from inside a submersible going off Norway to look at the coral reefs down there. And this to me captures a large amount of the challenge that we face. So this was a snapshot in time, a picture of this reef. We can see it's a really complex habitat, but just getting down to these habitats is very time consuming, expensive, and just very, very challenging. But the challenge for us is then how can we understand these ecosystems to predict what's going to happen to them in the future? And this is where the work I'll be talking about today is really going. So here's just another picture of a cold water coral ecosystem. So this one is off our west coast, more towards the Irish margins. And you can see here there's a really complex three-dimensional habitat. And what I want you to take from this picture is the amount of grey coral. This is dead coral, so exposed skeleton. So we have the white live coral and the orange live coral, but mostly grey coral. And this is a normal picture of a healthy, normal cold water coral reef habitat. It's mostly dead coral with some live coral on top. But of course, the dead framework is creating a lot of habitat for all sorts of different organisms. So one of the main threats to these cold water corals, and in particular the dead coral skeleton, is something called coral porosis. So we have osteoporosis, so porosis of our bones, osteo, but this is going to be porosis of the coral skeleton, so here coined coral porosis. And an important point, so James mentioned in his talk about the potential of adaptation of the pteropods and foraminifera, but with the dead coral skeleton, once it's there, there's no chance for adaptation. So this is going to be impacts on the dead skeleton with no chance for adaptation going forward. So here in this image, we have a slice of a cold water coral reef in the middle. And on the right-hand side, we can see some close-up pictures of that going from top down a little bit deeper. On the left-hand side, we've got a little bit of the chemistry. And through the middle of this, you've got this dashed line, which is the aragonite saturation horizon. Now, aragonite is the type of calcium carbonate that these cold water corals build their skeletons out of. Above this aragonite saturation layer, it's stable, there's no dissolution, but below it, exposed calcium carbonate, so dead coral skeleton, can start to dissolve. Now, the challenge that we have is that this aragonite saturation horizon is going to get shallower and shallower and shallower. So by the end of the century, the majority of cold water coral habitats are going to be sitting below this aragonite saturation horizon. Now, if we look on the right-hand side, we can see at the top where the calcium carbonate is stable, you've got this normal reef picture, a mixture of live and dead coral, lots of habitat provision. As we go a little bit deeper to the ash, and then even below it, we start to get this coral porosis. The coral skeleton, the dead coral skeleton, starts to dissolve. It gets unable to hold the reef above it. It collapses down, it crumbles, and you start to get a much simpler habitat, which is going to host a different type of biodiversity than the normal reef picture, which we saw before. So here's just a picture of a cold water coral, just so you can see the line between the live cold water coral and the dead one. So under normal circumstances, you've got this live tissue, which is protecting the skeleton. But as this coral grows, parts of it will die, the tissue will retract, and you end up with this exposed skeleton. But nonetheless, this is the foundation of these reefs. And here you can see towards the bottom right, this tissue starting to retract, this skeleton starting to be exposed. And if we zoom in on that, so this coral was from below the aragonite saturation horizon, and you can start to see the impact on the skeleton, which was not covered, not protected by the tissue. And you can start to get this porosity, this etching, this loss of calcium carbonate mineral. So how can we start to quantify some of this coral porosis? Well, first of all, we can go in situ. So inset, we can see the picture which I showed you before, but the larger picture here is from a cold water coral reef off the west coast of the States, where there's a deep water upwelling. And so even though the reefs are quite shallow, they're living in aragonite undersaturated waters, waters which are likely going to be experienced by most cold water coral reefs in the future. And what you can see here is that the dead coral skeleton is largely absent. This is no longer in this picture. So what we have here is live cold water coral living on bare exposed rock. So you've lost a huge amount of that habitat complexity. So still a reef ecosystem, but very different from the one which I showed you before of the Irish margins. Now we've also got lots of ex situ or experimental evidence where we've removed some live cold water coral, we've exposed them to long-term experiments. So these are year-long experiments, and we've done a few of those now. And we subject the live coral and dead coral to future ocean acidification scenarios. And importantly, this gives us timescales of what ocean acidification occurs over. So we can start to see the onset of coral process, and we can understand the timescales of this. And we've been able to take samples both from in situ and ex situ experimentation down to the Diamond synchrotron, Diamond Light Source synchrotron facility down in Oxford. And so you may have heard of the CERN Large Hadron Collider, which is a 26-mile long ring. And this is the UK's slightly more modest version, which is a mile ring, but it still gives you really high-powered X-rays, which we can use to look at the skeletons. And here in the middle, this blue arm, this is the robotic arm, which was recently installed, which allowed us to take the little carousel. You can see all our coral samples at the bottom and load those into this very high-powered X-ray. And so this will give us images such as this. So here we have two coral skeletons from below the aragonite saturation horizon. The one on the left was living, so the skeleton was protected by the tissue, and the one on the right was where there was no tissue protecting the skeleton, and it was exposed to ocean acidification and dissolution. So on the left-hand side, you can really see all those fine surface features preserved. On the right-hand side, you can see chunks coming off it. You can see the spongy appearance of some of the outside walls and just loss of some material in general. If we take those two same images and this time go down through them, on the left, you can see that there's really not a lot of porosity, no holes in the skeleton. On the right-hand side, you can see this spongy appearance coming around the outside of the wall. And this is this coral process in action. Now, you may look at this picture and think like, well, it's only a little bit around the outside of the coral. But from working with colleagues who are material scientists and people that work in the osteoporosis field as well, so engineers, this is the structurally critical part of the coral. So dissolution around this outside edge makes it much more brittle, like it would do in our bones as well. And so here's some images from some of the long-term experimentation we've done. So going left to right, we've got a timescale, so going from 0 month all the way to 12 months. On the top row, we have our control corals in non-acidified conditions, and in the bottom one, we have some corals exposed to future ocean conditions. And these are corals which were repeatedly sampled over time, so this is the same individual coral. And what we can see that in the future conditions, you can see it's starting to get a little bit fuzzy around the outside, and you're just getting thinner and thinner walls and less material as we go over time. Now, if you think about this as the base of this massive structure, you can see why there'd be a problem. It's getting thinner. It's unable to support the weight of a reef above it. So working with some of these mechanical engineers and material scientists, it was very useful to go from my attempts at engineering and having a look at the breaking strength of the corals I did in my early papers. We worked with them instead, and then we started to go much more into the stress strain of all these individual corals. We can assume that these corals are a hollow beam, and then we can apply a lot of well-known, well-tried and tested material engineering principles to this. And so one of the things we found is that as we get a larger amount of porosity in these coral skeletons, we decrease the skeleton stiffness, which is the initial slope in these graphs, and we also decrease the maximum bearable mechanical stress, which is the inflection points on each of these three coloured lines to the right. So in a nutshell, as we increase porosity, these skeletons are getting weaker and unable to support the reef above them. So now we've gone from there to make the first attempts at an in silico model, and where we can really model the mechanical strength and the structure of these individual corals. Now, we've been able to do this by taking a lot of this in situ and ex situ evidence, which we've got by looking at all these timescales of porosity and the amounts of porosity using the synchrotron samples. And we've come up with a nice model. But this is also limited in what we can do at the moment. So this is all based upon small coral samples that we've collected or experimented on as well. So this model works, but it's only to a certain side, which is not ecologically relevant at the moment. So the challenge for us now is how can we scale this up? And the current project, which we've just started, is looking exactly at this. So this is a snapshot of where we are and why we positioned the current project where it was. So if we think about the size scale, we're going from all the way from little fragments to single colonies to many colonies to thousands of colonies, which is the in situ environment. So we've got a really good handle on the small scale side of things. So we have existing data on coral porosis, the amounts of material which are lost, the mechanical properties of the corals. We've been able to resolve models which can give us the structural integrity of individual corals and various morphologies. We've scanned hundreds of different corals. So we've got good, highly resolved data there. If we go to the other end of the scale, when we have a look at in situ, we have existing imagery of reefs along an environmental gradient, so we can see what these reefs look like at different aragonite saturations, and we also can understand the amount of live and dead coral material. But one of the challenges from taking material from these environments is you don't know how long these corals have been exposed to different acidification conditions or the extent of the acidification conditions as well. So a few gaps with that. But what we need is something in the middle where we can take something ecologically relevant and experiment on this in the lab, but under timescales where we can start to see some changes, which is currently not possible in the lab, which is why we thought we needed a bit of a change in how we can approach this. So to do this, we need to be able to visualize this habitat loss in the lab. So we need to speed up this process, so over weeks and months rather than decades. So if we were to just do this ocean acidification experiment at the moment, we would have to wait decades to see anything happening, which of course is not feasible. We also need to perform these experiments at a scale that's ecologically relevant. Now, these are two things which many of you will be thinking, well, how is this going to work? And the way that we're going to do this is we're going to 3D print corals made entirely out of calcium carbonate, so no plastic polymer within that. And this will allow us to make replicable units which we can then print multiple miniature reefs and put these into these different accelerated conditions. And of course, there's lots of material validation that we need to perform as part of this because it's not biologically produced calcium carbonate, but it's produced by the 3D printer. But this allows us to 3D print these corals, make replicable units, and it also allows us to make these miniature reefs. We can miniaturize this whole reef process. So to make it environmentally relevant, we have shrunk the reef size and we're making it faster. And we can only do this, though, because we've spent a huge amount of time already with the ex situ work and the in situ work. So we know the real scales and the real rates of all of these processes. So this process has only started a few weeks ago. So these are just some test pieces of the 3D printed calcium carbonate corals. And just the pictures on the right just show you something which was probably about the size of my head, now shrunk down to a millimeter or 2 centimeters across. So this is the scale that we're reducing the size of some of these reefs. And so as we bring that together, then we can create these 3D miniature reefs. Then we can expose them to the accelerated acidification conditions. Now, in the middle top, I've got some orange and some grey corals. Now, this experimentally represents the living coral fraction and the dead coral fraction. And to simulate the tissue coverage, we've been trialing paraffin wax coverage, and we did some experiments last year, which we're just writing up now, where we can cover corals or 3D printed corals with wax to simulate this tissue. And this does work very well to protect that tissue from ocean acidification effects. So we can replicate this in the lab. We will then have a look at how the structure changes over time using photogrammetry techniques, and I'll show that in the next slide. And we will also do CT testing of the entire structure before and after, so we can see that loss and calculate that loss of habitable volume as well. We'll also do various mechanical and structural tests on these whole reef frameworks as well. Then we can bring this into our existing reef in silico reef model to expand upon this, and we'll be using this with the existing in situ and also the ex situ evidence as well. And going to this one, so we set up our new photogrammetry rig. So this is just a screenshot of my screen, which I did. We've only just set this up in the last few days, so I don't have a slick model of this yet, but this will give you the idea of the kind of photogrammetry which we can do. This is just by using phones and just the LiDAR sensors on our phones now. So one of the things which we're quite keen to do as well is we'll be testing some much more expensive equipment, but also with very accessible LiDAR equipment through our phones as well, to try and really make some methodology which will be useful going forward for the community as well. So this is some of the photogrammetry which we'll do to see that loss in habitat as we go through the experiments, but we'll be supplementing this with the full CT scans before and afterwards as well. So just to bring some of this together, what we have is long-term data on real corals exposed to various future scenarios. So we've got the ex situ data from the long-term experiments. We have the in situ evidence, so we know what the future will look like, but we've also got real samples, and we can see how that coral ecosystem changes along an environmental gradient. We've already incorporated this into an in silico model, but what we need now is to scale this up into something ecologically relevant. So we're doing this by 3D printing these replicable miniature reefs for testing various ecosystem settings, such as the amount of live and dead corals. As we saw from the earlier pictures, some reefs are mostly dead corals with a little bit of live corals, but we'll test different settings within this. So maybe more live, less dead coral as well. We will also do this for different ecosystems. So on top of the seamount, slopes, canyons, we're interested in looking at all of these different coral ecosystem types. So coming towards the end of this project, but also beyond this, what we really want to be able to do is to start to create some risk models of this habitat loss of these vulnerable marine ecosystems, and ideally something which is user interfaceable, where you can put in the amount of live and dead coral which you have, go region by region, look at the more specific environmental projections for that region, and get towards whether there is a large risk of ecosystem loss or that habitat loss from the cold water corals. And then, of course, then that needs to fit into all sorts of other questions like, well, what does that actually mean in terms of the biodiversity supports, and what are the timescales? Do they match, or is there a mismatch? Plenty of other questions, but in a nutshell, we want to go towards a risk model of habitat loss with all of this. And I'd just like to say thank you for listening, and just finish with a picture of a coral. And you can just see, this is one of the experimental corals, and you can just see the really sharp line where there was still some tissue on the coral and where the dead skeleton outside was exposed to ocean acidification. You can really see that loss of material. So I'll say thank you very much, and I'll leave that there, and I'll stop sharing my screen.